Okay, so welcome and thank you everybody for joining us today for another session of the PCE3 seminar series. And if you didn't know, PCE3 stands for Prebiotic Chemistry and Early Earth Environments. And one of the main goals of PCE3 is to bring together scientists from various disciplines to work together towards better understanding the origin of life. And having said that, we kind of want to get an idea of what everyone's research interests are. So we're going to put up a short poll. I'll put that up now. And I guess as that's going up, I'll just have a few more words. And so we're excited today to have two early career researchers, and we'll be hearing from Qiyuan Zhao from Purdue University, and also Bruno Cuevas Zuviria from the University of Wisconsin-Madison. And one quick note, we'd like to keep the chat closed during the talks, and we'll open up the chat to Q&A at the end of both talks. So please try to make a note of any questions that pop up during any of the presentations. And so, let me close this poll. So first, we're going to be hearing from Qiyuan Zhao, who's a PhD candidate at Purdue University, working with Professor Brett Savoy with a focus on innovative approaches to reaction network exploration. And today he's going to share his recent work on the comprehensive exploration of prebiotic reaction networks using a novel model. And so, Qiyuan, if you're ready, you can go ahead.
Sure. Let me share my slides.
Yeah, that looks great.
Okay, cool. Cool, great. Good afternoon, everyone. Thanks for the kind introduction. And it's my great pleasure to be invited to give a presentation about the prebiotic chemical network. As been introduced, my name is Qiyuan Zhao, a fifth-year PhD candidate from Purdue Chemical Engineering Department, and my advisor is Professor Brett Savoy. Today, I'd like to talk about our recent publication on thermally accessible prebiotic pathways of forming RNA and protein precursors from aqueous hydrogen cyanide. Speaking of the origin of life, one popular hypothesis is the RNA world hypothesis, which proposed RNA played a crucial role in the origin of life on Earth. The hypothesis suggests that before the evolution of DNA and proteins, RNA serve as both the genetic material and the catalyst for biochemical reactions necessary for life. Among various of simple molecules, aqueous hydrogen cyanide has been proposed as a possible precursor to many of the important biomolecules in this process. Starting from 1950s, experimental studies has been conducted to test the hypothesis that the building block of the life could have formed spontaneously on the early Earth, providing the foundation of the development of life. So as being highlighted in this panel, we can observe that various of sugar, RNA, and protein precursors has been identified experimentally by analyzing the reactions happened in the aqueous hydrogen cyanide. However, experimental studies focus more on the stable and final products, while the detailed reaction pathways and intermediates can be relatively hard to capture in experiments. And with the maturation of quantum chemistry methods starting from last decades, computational studies has become more widely applied to investigate the possible reactions in the aqueous hydrogen cyanide system, and has reviewed many detailed reaction mechanisms. Like the two studies shown here, computational chemists use their domain knowledge to hypothesize possible reactions. Like here, they hypothesize possible reactions of forming formamide dimers and trimers. And then they use quantum chemistry methods to test their hypothesis to establish transition state and reaction pathways and other important properties. More recently, instead of these ad hoc and domain knowledge guided approaches, the development of the called AB initial nanoreactor allows for automated reaction exploration and has also been commonly used to study the prebiotic chemistry. Like in a recent study, researchers put 15 hydrogen cyanide and 16 water molecules in a nanoreactor and provide extreme condition, like extremely high temperature and pressure to push the molecules to react with each other and speed up the reaction process. They are able to discover many of the important precursors. So as been summarized in this right-hand side panel, although the domain knowledge guided exploration and the AB initial nanoreactor approach, they can achieve a relative deep reaction network exploration, as shown in this. Let me change it to either point. Yeah, highlighted by these red circles and red lines. They represent these sampling heuristics can allow you to go deep and find those important products. However, it cannot achieve a comprehensive reaction exploration. Considering the scenario and showing this gray circle here, if we can perform a comprehensive search, it is quite likely we can find some new intermediates, some reactions with lower barriers compared with sampling heuristics. This can be pretty important for the reaction network, because you can imagine if some intermediates and reaction pathways are missed at the beginning of the reaction network, the kinetics and the evolution of the entire network can be inaccurate. So this leads to today's topic. So as a non-specialized chemist who is new to the field of prebiotic chemistry, is it possible to establish a comprehensive reaction network and discover some new unreported interesting chemistries? So this leads to actually my PhD effort. One important focus of my entire PhD study is to establish a general solution for automated reaction prediction. And we developed a package called Yet Another Reaction Program, or in short, call it YARP, to achieve this goal. So the key idea of YARP is turn the A to question mark problem. A means the starting reactant. We only have the information of the reactant. We don't know anything about the intermediates and the possible reaction in this system. And we turn the A to question mark problem into tractable and solvable A to B problems. To do so, we first use a graph-based product enumeration instead of doing a random or brute force potential energy surface exploration. We turn the reactant species into chemical graph, and then use a graph-defined elemental reaction step defined by the number of bond changes. For example, in the neutral and closed shell system, we prove the simplest reaction step is the breaking two bonds and forming two bonds reaction. It will lead to another closed shell species. And in this way, you can see that we only need to care about number of bond changes. We don't need to care about which bond is easier to break or not. So it requires limited demand of chemical heuristics, which also allows it to be easily applied to an arbitrary new system. And after enumerating those products, we take advantage of recent developments in semi-empirical methods. Like here, we show it's a method called TSIT, Tophic component theory, to quickly estimate the thermochemistry properties to pre-proning those reactions and enumerate products. And we also use semi-empirical quantum chemistry as well as those machine learning force field, like semi-empirical quantum chemistry, we use GFN2XTB, and for machine learning force field, sometimes we use those NA or MNet force field. We use this cheap method to explore the potential end surface and to generate possible reactions. And after that, we use a more accurate density functional theory to refine those transition state and get more accurate properties like activation energy and enthalpy of reaction. And after each step exploration, we will get many different new intermediates, new products. We can select those more important ones based on the activation energy. Like we can set an activation energy threshold and we only care about those low barrier reactions. We name it as kinetically accessible intermediates. We use it to seed for deeper reaction exploration. So you can see recursively using this process, we can eventually generate a deep reaction network. In one of its benchmarking case, we applied YARP to study the decomposition of the gamma keto job site, which is an important material for a combustion system. We show that YARP can automatically identify all those previously established reaction pathways and intermediates, and it can find more. It can find many new products and new lower barrier reaction pathways. More importantly, we show that YARP framework can increase the exploration efficiency by 10-fold while decrease the computational cost by 100-fold compared with other similar methods. So it allows us to do a reaction exploration from scratch in the prebiotic chemistry system. In this study, we apply YARP to comprehensively enumerate all the unimolecular and bimolecular breaking two bonds, forming two bonds reactions involving water, hydrogen cyanide, and the product, the intermediates, out to a depth of 4. And we show that even just for a relatively shallow reaction network, a broad chemical space has been established. And I will break down the network based on several key observations. So in the beginning, the first reaction step is water and hydrogen cyanide to form formic acid. And this formic acid can later easily transfer it into a more stable isomer, the formamide. And it can also react with water to form a aminodiol species. And all these two species can contribute to the formation of formic acid, but with quite different activation energy. Like going from formamide, the activation energy of this transformation is 47 kcal/mol, which is pretty high. But if you go through this aminodiol species, the barrier can be reduced to 15 kcal/mol. Continue the exploration, another important species, carbon dioxide, has been discovered. And again, we show the reaction exploration find two alternative reaction pathways. One is going from the aminodiol species. It first decomposed into carbonic acid and then decomposed into carbon dioxide and ammonia. The activation energy of this of these two steps are around 40 kcal/mol. And alternatively, you still go from the aminodiol or from the formaldehyde intermediates. You can first form formic acid and then do a hydrogen elimination to form carbon dioxide with relative high activation energy. We'll compare those two reaction pathways in more detailed way. And another important intermediate I want to highlight here is the methylene amine. So here we can show that in our network exploration, it can be easily formed through 3-step reaction. However, in another approach, someone used the ab initio nanoreactor. They also established this intermediate, but from a quite different and quite complex reaction mechanism. It in total needs 6 steps, and some steps is pretty high activation energy to finally generate the methylene amine. So here we can see how a comprehensive reaction exploration can find new reaction pathways, and the importance of this methylene amine intermediate in the whole network is hugely underestimated by previous approach. And continue the exploration. Another interesting observation of the network is the backtracking reaction. So here we show these intermediates. It can be generated from other reaction directions, and we perform a unimolecular reaction exploration on it. We find 3 decomposition pathways to forming 3 combinations of formic acid and formamide. However, the arrow here only represents the certain direction when we apply YARP. Considering the high concentration of these 2 species, when we perform a microkinetic modeling, we actually show the reaction flux is reverse. These 2 intermediates generate going through a bimolecular reaction to generate intermediates, which further decompose into formic acid and formamide. So to briefly summarize this reaction network, we can see a surprisingly diverse reactive landscape was reviewed with just a few steps of these simple molecules. And one way to simplify the complex reaction network and highlight more important reactions and intermediates is a microkinetic modeling. I mentioned it's in some of previous slides. So considering an elementary reaction, which have 2 reactants A and B and 2 products C and D, the kinetic, the reaction rate can be calculated based on some transition state theory like Aaron equation. So if you know the activation energy, which is already obtained in our reaction exploration step, you can calculate the reaction rate and perform a kinetic simulation. So considering a simple scenario, you have a sequential elementary step starting from reactant R, you go to 3 intermediate A, B, and C, and eventually go to P. You can write down 5 differential equations. You can simulate the change of concentration by the rate, by the concentration of reactant and the rate constant. And similarly, for a complex network, you can always have a set of dependent differential equations. You can solve it numerically given an initial condition. You can know the evolution of the entire reaction network. So here we summarize 20 reactions with the highest reaction flux, which to simplify the original reaction network. So some key observation is that we notice the formation of important species like formic acid and carbon dioxide. They are all formed. And as mentioned before, following the microkinetic modeling result, the reaction flux is going from formic acid and to these key intermediates. So all our previous hypothesis has been verified. And in the following slides, we will analyze the reaction pathways towards some important product more detail by 2 metrics. One is the relative energy and the other one is kinetic importance. So first we talk about the reaction pathway of formic acid formation. We first list 2 reaction pathways. They are all identified by our comprehensive reaction exploration. And the red one is also reported by the previous study, nanoreactor study, which shows that the red curve here need to go through a relative high activation energy transformation, 47 kcal per mole, to go from formamide to formic acid. However, for the black line, if we introduce a new key intermediate aminodiol species, the overall barrier, which is defined as the highest activation energy among the sequential reaction pathways, it just describes the rate limiting step. It can dramatically reduce the overall barrier and improve the kinetic preference of this reaction pathway. As mentioned before, we also have the biomolecular reaction, the backtracking reaction identified in this search. These 2 intermediates forms the other more complex structure and further decompose into formic acid. This is also have lower overall barrier compared with the previously reported one, so we call it alternative kinetically favorable pathway. More importantly, as we perform the kinetic modeling, we are able to track the concentration change of each species, and we can also track which reaction pathway contributes most to the formation of formic acid. Here we call it the pathway with the highest reaction flux. Surprisingly, this reaction pathway with the highest reaction flux is none of these 3. It's actually the blue line shown here, which connect 2 of the key intermediates formamide with aminodiol species, connecting those 2 to contribute to the formation of formic acid. The rationale for this is if you compare the black line, which has the lowest overall barrier, and this blue line has the highest flux, the key difference is whether the reaction go from these key intermediates or from the formamide, because the extremely low energy, which means this one is more stable than the previous one. It has a huge concentration deviation. The concentration deviation can be large to order of magnitude 8 magnitude. So this explains why the blue line contribute most to the formic acid formation. Similarly, we perform the analysis for the carbon dioxide formation, and we still get 2 reaction pathway. One is the lowest overall barrier one, and the other one is previously reported one, and they are all identified in our study. And similarly, the pathway with the highest reaction flux is this blue line. It's still connecting it goes through from formamide to this aminodiol species to connecting this to connect the red. It's slightly different from this red line, but it has the highest concentration. So 2 key observations here is the first one: if you only compare the relative energy, like if you only care about the overall barrier along the reaction pathway, it can sometimes be misleading because you ignore the dynamic of concentration of each intermediates, which can lead to a wrong conclusion. And also our comprehensive reaction exploration discovered both the energetic and kinetically more favorable reactions compared with previous sampling heuristics. So it actually verified our first hypothesis. If you just sampling heuristic can easily miss the some important reactions and intermediates. Another interesting benchmark of our reaction network can be obtained with the comparison of an experimental study. So here, in an earlier experimental study, researchers performed a mass spec analysis and hypothesized some reaction mechanism. Like one of it is a autocatalytic loop. They hypothesized the triazine serve as a source of hydrogen cyanide, and it's decomposed into 3 hydrogen cyanide, and the hydrogen cyanide reacts with water to form the formamide. And this is actually captured by our network exploration. And it's also the hypoxia reaction pathway miss one step. It's actually need a keto tautomerization to form the formamide. It has excuse because mass spec cannot tell the difference between those similar isomers. So this also shows the advantage of the reaction network exploration. It can capture all those key intermediates and isomers. And once it forms the formamide, it continue react with water and hydrogen cyanide to form 2 intermediates. These 2 are also captured in our exploration. I highlighted by this red circle and blue circle. And we also find corresponding isomers, which again cannot be hard to be predicted by the mass spec analysis. And one key difference is the hypothesize once they form this these intermediates, it decomposes into 2 formaldehyde. But as we mentioned before, if we're considering the kinetic modeling, we're considering the concentration effect, the reaction direction is actually wrong. It actually should be flicked back. These 2 formamide contribute to these intermediates and further decompose into other products like ammonia. So we can see from this comparison, it's not a direct comparison, but we can see this comparison verify the existence of key intermediates proposed by experimental study and also identify corresponding isomers, which can hard to be detected. And our simulations suggest different reaction direction and different reaction evolution in the reaction network. So far, all those reactions, the reaction network we talk about is in the neutral closed shell system. But I want to highlight here our reaction exploration, or YARP, can not only deal with these closed shell species, it can also be extended to radical and charged species. So here we show a 3-step reaction network with the presence of hydroxyl radical. Although we can see it's have a different energy landscape, like with the presence of hydroxyl radical, which is more reactive. The activation energy of many reactions is lower than the previous one. So this topic is not to want to compare like radical reaction network and closed shell reaction network. I just want to highlight that in some cases, if it's necessary, our reaction network exploration can involve those radical and charged species as well. In the end, I also want to highlight some additional benefits of our approach. In previous slides, we showed that our approach can comprehensively capture many important and new intermediates. It has some other benefits. First is it needs much less computational cost. So here we counted the number of gradient costs, which is a rate limiting step. It's required to do the geometry optimization and the transition state optimization. We break down it into different layers. So for the layer 1, we just need 54 gradient costs, and layer 4, we need 5,000. Summing them all together, we need around 8,000 gradient costs. So this value is hard to make any sense. So we also counted the number of gradient costs needed for running an AB initial nanoreactor simulation. So in the same system, if you want to run an AB initial nanoreactor simulation, you need 1 million gradient costs for one trajectory. Sometimes people need to use different 10 to 20 different trajectories to finally get a relatively entire reaction network. Although it's like a orange to apple comparison, because one may argue you are using 2 different levels of theory. You are using an expensive one, but nanoreactor are using a cheap one. But it's still reflects that our reaction exploration is more efficient. And another benefit is that we can have some more systematic consideration of different effects. Like here in this study, we consider the explicit solvation effect. For every enumerated reaction, we always consider if we add in a water serve as a proton shuttle, if this water catalyzed reaction can reduce the activation energy or not. Like, for example, for the keto-enol tautomerization here, adding additional water serve as catalyst can dramatically reduce the activation energy, typically by 20 to 30 kcal per mole. And with this, we can be more accurate to generate this comprehensive reaction network. Okay, so brief summarize today's topic. Our study established reaction network of water and hydrogen cyanide, which identify many important building stock of prebiotic chemistry. And the discovery of kinetically more favorable reaction pathways illustrates the necessity of comprehensive reaction exploration rather than just sampling heuristics. And as automated reaction prediction and its necessary modules continue to mature, I believe they offer a potential for a wide range of applications. As for the outlook aspect, I believe the deeper and more complex reaction networks, now we're just going to depths of 4, and more complex reaction network can be established with the use of a exploration policy and some machine learning tools to further speed up the transition state calculation. And the interaction of computational reaction networks with experimental studies will become even more diverse and complementary. They can be compared in different aspects. So before the end of this talk, I want to highlight some ongoing project or more recent project we are doing to push forward to the outlook. So here in our recent study, we developed a reaction exploration policy. We name it as a modified Dasher algorithm. It can control the searching scope while also keep track of the most important intermediates. So with that, we apply to a process of biomass system. It generates a very deep reaction network. It connects the initial reactant with all those experimentally identified major products, and it can even accurately predict the relative yields of each one. And meanwhile, we also develop machine learning models to speed up the activation energy prediction. So here we develop a model called Dirt Square model. You just need to do everything at semi-empirical quantum chemistry level. And the model will learn the difference between XTB, the GFN to XTB, and the DFT level theory. It can perfectly reproduce DFT, and sometimes if you give it some new data, give it 1% of data of higher level theory, here we use Gaussian 4, you can consider it as a golden standard method. The Dirt Square model can even outperform the DFT. So all these two means that we are now able to do more complex reaction network exploration in the prebiotic chemistry. So I'd like to thanks for my advisor, Professor Boy, and thanks for this Boy research group, and also thanks for those junior students helped me to finish these projects. Also thanks for our collaborators from Carnegie Mellon to set up these machine learning models. And also thanks for the funding source, and thanks for your attention.
Great. Thank you so much for sharing, Qiyuan. That was a very interesting talk. And so if anyone had questions for Qiyuan, we're going to save the Q&A till the very end. So please hold on to your questions for him. And for now, we'll be moving on to our second presentation. And that is going to be given by Bruno Cuevas. And Bruno Cuevas is a postdoctoral scholar working at University of Wisconsin-Madison, and he received his PhD from the Universidad Politécnica de Madrid, where he studied machine learning enabled prediction of electron densities. And now at the University of Wisconsin, he's working with Professor Betul Kachar, and he received the Margarita Salas postdoctoral fellowship, and his main interests are in computational modeling of the evolution of chemical reaction networks and the evolution of protein dynamics. And so whenever you're ready, go ahead, either should be ready to go, Bruno.
Hello?
Yeah,
Great.
yeah, we can hear you. Excuse me.
Okay, great. Hi, everyone. My name is Bruno Cuevas, and I'm a postdoctoral researcher at the lab of Betul Gashar at the University of Wisconsin-Madison. And I'm also a Margarita Salas Fellow at the Universidad Politécnica Madrid, as I was introduced. So thanks. I want to thank the organizer. I'm very happy to have this chance to talk today at this forum about one of my favorite topics, reaction networks, and about how these reaction networks might be related with the origin of life. So when we study life in general, we find that networks at almost every level. So if we go for the molecular level, we'll find that protein structures make networks that allow us to explain certain phenomena, like, for instance, allosteric signaling. If we go to the cellular level, we'll find that cells have all kinds of genes, enzymes, and metabolites connected with each other, making the metabolic networks or the gene regulation networks. And if we go to the ecosystem level, to the ecological level, we'll find that every species on an ecosystem will be at some point interacting with other species, either by parasiting or either by cooperating or predating. So in the end, we find like a networking at every level. So this could be an anecdote or a coincidence, but actually I think that this is something else. And so far, like the characterization of like all the literature that we have collected until now might indicate us that networks are a fundamental part of life, and that some of the properties that we find in living beings can only be explained by understanding these networks. So the same way that we need networks to understand life, extend life, it's also possible that we will need to use networks to understand the origins of life. So actually, indeed, this has been proposed by earlier work. There has been earlier work that has highlighted how relevant are networks for the research in the origins of life. So, for instance, here we can see that work in the early 2000s show that the metabolic networks that we find in extant organisms, they are organized around heavy tail degree distributions. That is a distribution that we find in many real systems, as for instance, the internet, transportation systems, etc. Some later work, for instance, had a look at the extant metabolic networks to find some fossils that might be embedded there, as for instance, the case of metabolism without phosphate that might be hidden there on our extant metabolisms. And also, there are like experiments that are using ideas borrowed from network theory to explain the evolution of to explain certain evolution experiments. On the other hand, on the other side of the origins of life research, from a more like bottom-up approach, we have that networks have been used to explain certain, for instance, to explain the pathways that could lead us to the generation of different biomolecules in different prebiotic scenarios. Networks have been also proposed as a way to explain like how different chemical reactivities might have properties that are more similar to certain properties of living organisms. And also there's been recent work that has proven how chemical reaction networks can be tuned with environmental conditions to generate specific products in the absence of catalysts. So today I'm gonna talk about a couple projects that they are both in these two directions, in the bottom-up approach and on the top-down approach. So I'm gonna start with my project involving prebiotic chemical reaction networks. So when I arrived to this field 1.5 years ago, I was very surprised by finding like how much we learned in this 70 years after the urine meal experiments. For instance, in this case, I'm showing a figure from a 2018 article, and here this is covering some part of the prebiotic chemistry that was known until then. And here we can see that in the end, we are able to generate from prebiotic precursors many of the different biomolecules that we find in living organisms. So far, this problem is not solved, and this is still like an ongoing thing. But when I look at this, I feel like both fascination and overwhelmed, because in the end, there is like a lot of there are like a lot of chemical reactions, and not only those chemical reactions, but the interaction of those reactions and environment, which is a very important part of prebiotic chemistry. So since I landed here, I've been always wondering, like, okay, shouldn't we have like an easier way to work with this? So the way that I see it, in the end, this field is not new. Sorry, it's around 70 years old. So this means like there has been a lot of previous work. Then it's also a field that is not easy to navigate. If we consider that in the end, most people, we get our training in either chemistry or biology, but in the case of prebiotic chemistry, you have to understand a little bit of geology and a little bit of organic chemistry. So it's usually quite multidisciplinary. And also a field with some cultural factors, some cultural divides across different possibilities. So in the end, I think that these different problems, they might have a common solution, which is integrating the past, the present, and the future of prebiotic chemistry into the same place. And that place is something we can call a database. So I think if we built a database with what we know of prebiotic chemistry, we could really advance forward in this field. So I'm currently working on the creation of that database. So here I'm showing the current status of my project. This database will be fed by two kinds of information, like information that I will obtain from other databases that have open information, and also from the manual annotation of prebiotic chemical reactions. This manual annotation will come either from experiments or from the literature, from curating the literature. This is usually a process that is usually a little bit, annoying, a little bit painful. So I'm working on generating like apps and to annotate and curate data here. So it will be easier for scientists in the field to include information here. And then the data will be stored on a database. In this case, I'm using the Arango database, which is an example of a multimodal database that will allow us to store data as graphs, tables, or documents. And finally, that database will be available for users through a web interface or through a REST API. Those two are still in development. So the way I see it, like in the end, chemical reactions, they are connected. They generate these chemical reaction networks. So it seems natural to store information using more or less the same formula. So in this case, I'm showing you like a multipartite graph that represents like the different things that we can find in our efforts to store like prebiotic chemistry. And one of the good advantages of using such a graph approach to store this data is like complex questions, like for instance, what biomolecules could be elaborated from cyanide and sulfide, or what is the shortest pathway to generate a certain molecule from CO2, or which are the molecules that don't have any connection into them, can be easily solved with this by storing data on a network and performing like by performing queries in this data using network tools. However, the total amount of effort that will be needed for me to generate this chemical reaction network is, this database about prebiotic chemistry is far way more than I will have in my whole life, I think. So in the end, like for this project to succeed, it should turn into a community effort where like different scientists with different expertises will give a little bit to the field by curating and annotating data. So far, my current goal is to generate like a skeleton of chemical reactions that span different fields and that also include like as I say, a core, some base knowledge of chemical reactions in prebiotic chemistry. So from this starting point, we can start building on top of it. I think also it will benefit a lot the field to have like a resource that is also gathering a little bit of information from adjacent fields, as I included here, like atmospheric or atmospheric chemistry or geochemistry, because in the end, like the origin of life doesn't really understand about fields or about disciplines. So if we follow this way, we'll have a little bit of a broader approach. So just by putting all reactions together, we can actually have we can start already like trying to understand these systems. For instance, in this case, with the information that I collected so far, I'm trying to analyze some of the properties that the chemical reaction networks that we will find in these systems have. So, for instance, one of my favorite ones is like the way that how these systems are how the components of the systems are connected. So, as you can see here, we can find like clear differences between atmospheric chemical models and and biological/organic chemical reaction networks. Also, this approach will allow us to, as I said, to generate this, to understand prebiotic chemistry, but from a network perspective. So, for instance, here in this case, I'm showing a chemical reaction network that I built from different sources. So in this case, we are considering like a cyanosulfidic and a hydrothermal vent. I don't think that the system that we have a system that works like this. But anyway, just one of the things that I like about networks is that just by visualizing networks, we can understand some of the things that are happening here. So without requiring like any complex analysis. So for instance, in this case, we can see that there are like some molecules like alanine and glycine that are produced by by the different models of this network. However, like these models also have a different, they get to these molecules through different pathways. So for instance, we can see on the left that the cyanide has like a central role in the around cyanosulfidic chemistry because it's the main carbon feedstock. While on the case of hydrothermal vents, we will find that CO2 has that role. So one of the catches that we have in this, how we say, literature-based approach to chemical reaction networks is that in the end, these chemical reaction networks are usually biased towards reporting the synthesis of biomolecules. So if we just look at those, we will have at the end the feeling like producing biomolecules is easy. And actually, every prebiotic chemical setup is very likely to produce many side reactions that might also play some role in the evolution of the origins of life. So one thing that we can do, even in that direction, this prebiotic chemical reaction database can help. For instance, in this case, I'm considering like a set of rules, of chemical reaction rules, which is something that has been already employed to characterize prebiotic chemistry. But in this case, I'm fitting, besides performing this expansion based on chemical reaction rules, I'm also using machine learning tools to try to fill the gaps, try to include like the reactions that I didn't come up with. So in that way, we can generate like richer chemical reaction networks with more intermediates and more species than we could do before. So in this case, getting like a comprehensive collection of prebiotic chemistry can help us to train these models and to be more accurate in our domain. Something I forgot to say is like right now I'm calling it PrebChem database. I'm pretty terrible at choosing names. So if anybody has a better name than this, I will be very happy to hear it because I'm not very good at that. So right now this is a current work in progress. I hope it can be released soon. And now I'd like to talk about my other research topic, which is also related with reaction networks, but in this case, it's related with networks from the other side. In this case, we can talk about like the top-down approach, so analyzing the external life to try to find something about its origins. So in this case, I'm feeling like especially attracted to the idea of translation, to the process of translation. So there, in the end, the origin of life field, it has like endless catch-22 situations due to a reducible complexity of life. Like for instance, we all know that we can extract energy and matter without metabolism, and we can do metabolism without enzymes. So in between these two, between these two processes that are a bit like say entangled, we find translation, and translation is a quite remarkable process. It is an information decoding process that involves a megadalton molecular machinery that has no known counterpart on the natural world, and whose emergence was described as one of the greatest mysteries in the origins of life puzzle. Therefore, all organisms have one, and despite all the billion years of divergence of this machinery, it has stayed fundamentally the same. So, in order to understand this central system a little bit better, we decided to build a chemosynthetic translation network of the molecular biology best known organism, which is E. coli. So, to build this network, my coworkers, Erin, Zach, and me, we had to check the state of the art regarding translation, covering around 100 articles and reviews. From what we learned from that literature search, we generate a set of reactions that we think that describe more or less accurately what happens inside translation. This is something that will always keep evolving. So it's something that we might change in the future, but so far, this is it. So from those reactions, we built a network trying to take care of these different aspects. Then we merged the translation network with a previously characterized metabolic network in E. coli. And we analyzed the results using standard graph analysis, network analysis tools. So here in this figure, I'm showing the schematic representation of the integrated cellular translation network. We built a cellular, our cellular network that includes, our translation network that includes all the relevant steps that we find in translation. That means initiation, elongation, etc. Then this layer, all these steps will generate the peptides that we find in the biosynthetic layer. Some of these peptides will give place to functional proteins by themselves, but some others will require the interaction with other proteins, with other peptides to generate this functional metabolic machinery. And then these proteins will interact with different metabolites at the metabolic level to enable either metabolic reactions or metabolic transport. And finally, in the metabolic layer, we will generate some of the building blocks and some of the energy molecules that will be consumed by the translation layer. So this integrated network spans all the cellular chemosynthetic processes, although it doesn't include explicitly all the processes like the genetic processes, the regulatory processes, or the compartmentalization of the cell. So here I'm depicting the translation network that we obtain. So as you can see, it's quite a scary network. It's quite dense. So the reason for this is we consider that in the end, the most important thing about translation is that it's at the process, decoding information process, which might resemble a bit like a computer. So in the attempt to model the informatic aspects of translation, we've accounted all the possible interactions between the different sites of the ribosome. So giving place to this network that has around more than 1,000 reactions. So given the huge parallelism of many of the reactions here, we can summarize, we can resimplify this network again to a little bit to what we are showing here. In the end, we have a big loop in which tRNAs charged with amino acids get in, and this loop and they get out in another part of the loop. And there are some control mechanisms here around the loop that allow the termination and the halt of the activity upon amino acid starvations. So this translation network is connected to metabolism. However, generating a chemical reaction network that contains around 24,000 nodes. However, visualizing that reaction network is a little bit cumbersome usually, so we decided to, in this case, we are showing a summarized version where we are reducing all the reactions that belong to the same model altogether. So what we can see here is in the end, despite these networks being necessary for translation being necessary for metabolism and vice versa, we still find that they are pretty modular, they are pretty separate, and actually the only things that keep them together are the energy molecules or the tRNA charging, as I'm showing here. So in this case, for instance, I'm showing the betweenness centrality of this network, which is the role of each of these nodes to traverse from one side of the network to the other. And we can see in general, like most of the tRNA charging is one of the most central, which is something that somehow we could expect. So once we have all this network, we might want to extract like some of the features of them. So as I say, one of my favorite features is of a network, and one of the easiest to analyze too, is the degree distribution. So when we have like the number of nodes that are connecting to each node. So in the case of these bipartite reaction networks that we are using, like the degree will be the number of reactions that in which each of these nodes participate. So we analyze this at the level of this overall network. We find that our network has a heavy tail degree distribution, which is something that we also find in other complex systems like internet, air traffic, or including metabolism. However, we also find that this heavy tail distribution is actually not the same for all the models that we have here. For instance, for metabolism, we already know that this was a heavy tail distribution, while in the case of biosynthesis, it's still a little bit ambiguous. Another property what we analyzed in this system was the energy distribution across different levels of the hierarchy. So in the end, like if we consider the cell as a level of nested hierarchies as genetics, translation, metabolism, etc., then we could expect that each of these layers, there is like an energy transfer process. So like the layer will retain some energy for itself and it will transmit whatever is left to the lower level. So in this kind of trophic chain idea of the cell, we will need a method to compute like what is the energy of each of these layers. And actually, we don't really have such a method, but actually we can use the molecular mass as a proxy of that energy, because in the end, in every biomolecular system, like the amount of energy that we have to devote to generate a specific to build a molecule might be proportional to the size of this molecule. So when we do that here, when we look at the mass, we actually find something that is a little bit shocking that we could expect from first principles. It's like in the end, there are like some mass gaps in these different layers, which might be indicating that something else is happening, because in the end, we will not expect this from a trophic organization of the cell. Luckily, this process this process could be just an anecdote, but actually there is like some evidence that this process also happens at the ecosystem level. So when we look at ecosystems, we will find that there are like some sizes for which we find no animal, and there are like certain reasons that might lead to this emergence of these gaps. So in the end, we wonder if we are not if this is not just an anecdote, but it might be a possible organizational principle of life. So I'd like to finish by introducing the Life RCN. So parallel to PC3, in this Life RCN was launched 1 year ago, and its objectives are also to spark the collaboration and the conversation between scientists. However, in this case, we are a bit more interested on the evolution of early life rather than the emergence of life to begin with. So in this one, we'll talk about other topics, maybe about, for instance, the emergence of multicellularity, the emergence of eukaryotes. So right now, I'm working with other people to create here a seminar program that will include all kind of profiles of multidisciplinary scientists, like paleontologists, microbiologists, etc. So stay tuned, because we might start like in this late spring. And with all of that, I like to thank all the people that I work with. Here as you can see, we are a big group and we're quite multidisciplinary. We work on a lot of projects, so here I'm highlighting like the people that directly contributed to this work in this case. And also like to thank all the institutions that allow me to do my research here, and also all of you for listening. And I will be happy to answer some questions.
Great, thanks, Bruno. And yeah, that's exciting to hear about the Life RCN seminar. I'm sure many people here would be interested to check that out. But for now, if anyone had any questions for Qiyuan or Bruno, the chat should be open, so you can type questions in there. And also, if you prefer to ask the question yourself, you could raise your hand in Zoom and we can unmute you. And yeah, so there's already a question in here from Uli Muller, and it is to Qiyuan, and he said, thanks for the great talk. And what pH did you assume for the reactions you modeled? Do you know how changes in pH affect the transition state energies and kinetics? And similarly, how sensitive is the network model to changes in water activity, wet-dry cycling?
Yeah, that's a very, very great question. So in our current study, we just considered pH equals 7. We didn't consider any acid effect, and yeah, so we did not systematically analyze the changes in pH affects the transition state. We just use the in-place solvation effect, use water as a solvent. So probably in the future we can considering more some different scenario and consider the changes in the water activity. Did that answer the question?
Okay, I think I saw that Kevin Devine raised his hand, so hopefully you're able to unmute yourself.
Yes, indeed. It's a question for Qiyuan once again, but thanks to both Qiyuan and Bruno for two very good, really excellent talks. It's a way of driving the field forward, getting a database where we've got all the information regarding all the multitude of prebiotic studies that have taken place over the last 70 years. It's 70 years exactly to 1953 since the first Urey-Miller experiment took place. And my question for Qiyuan, following on from what Uli asked about differences in pH, I think is very important. What about differences in ultraviolet flux? Because you mentioned about the effect of radical, especially hydrogen radicals, and certainly different levels of UV flux, at different wavelengths would certainly have a profound effect upon that. Are you also going to incorporate those into your models as well? I mean, there's a lot of other things you could do as well, but what about you?
Yeah.
Mm-hmm.
Yeah, I see your point. Yeah, it's another great point. So I have to acknowledge that because I'm new to the field, so we just start from the simple condition, like we don't consider the pH. We actually briefly mentioned in the last part of my presentation, I show a reaction network with the presence of hydroxyl radical, but the main effort is still focusing on those neutral closed shell species. So incorporating those more realistic experimental conditions I think it will be a pretty hard point to, let's say, bottleneck to connect the computational simulation with the experimental conditions. But I think our group will focus on that. We might have some, like to simulate the condensed phase, we will have some molecular dynamic simulations instead of just focusing on one molecule react with the other.
Because you could also, if you think about it, you could do it if you think of different environmental conditions, you could think of conditions around comets. For example, comets are loaded with organics that mostly in their lifetime very low temperature, but as they get close to the sun, the temperature increases, so does the UV flux. So to see what sort of building blocks are actually generated by these conditions, and then compare it with analysis of cometary material, I think this is very important, actually, because this determines exactly what molecular building blocks, what sort of like prebiotic precursors are delivered to planets, and in what relative abundances. I think so. These things I think are very important, that we get a good picture of this.
Yeah, get it. Yeah, thanks for this very important suggestion.
Thank you. Nice.
Great. I think so. someone else has their hand raised. So Donald Burke, hopefully you're able to unmute yourself now.
Yes, thanks for those two great talks. I'm so impressed by how the data science and computational approaches fields are adding new flavors to the analyses that we're doing here. As well as both Kevin and Uli for bringing up the question of conditioned space and how that might affect the outcomes. My question for you has to do with the math and the flux. When you're looking at starting from a single stage along the pathway, and then you have divergent paths you could take from there. I noticed that you said that one of your figures had a blue trace, and that was the one where the dominant flux was going through that path as opposed to the other one. That one, the material feeding through that path was generated by a step that had a very small transition state energy barrier. In getting from the common intermediate, then there was a small barrier to get to the next step, and then the blue path took over.
Let me share my screen maybe to help the discussion here.
And so I was wondering about the relative importance of the proximity of those low energy barrier steps. So it's not necessarily low energy barrier overall, but you can fill up some of the intermediates along the path by having low energy barriers to get there. And even though there no not this one, the stepping ladder that had the blue trace in it. Looking for the slide there.
This one or
Energy diagram.
energy diagram.
It was earlier, maybe.
Oh, I see. So this one.
Yes, right there. So TS2-2 is a much lower jump than getting to TS2-1.
That's true.
And so I would imagine that your compound that's shown as being minus 12.7, that would populate to a much greater degree than the black path that would have to take you over to 12.9.
Exactly.
But the very next step, going from minus 12.7 to plus 30.2, that's a 43 kcal jump. The blue path has a huge energy barrier, but you may be populating the material feeding into that path enough to overwhelm the high energy barrier for the blue path. So I guess my question for you, or maybe you can comment, can you comment on the relative importance of populating the intermediates as opposed to the high energy barriers that follow?
That's a very good point. So I think I gave some explanation when we're doing the kinetic simulation. Like when you're doing the kinetic simulation, when you compute the rate constant, it's not only related to the activation energy, which is the exponential term, it also associated with the concentration. So like in this case, we compare the concentration of the blue and this black line. Although the activation energy of the step is quite different, the population of these two can be various. The difference can be as large as to 10 to the 8 magnitude. So that's why the kinetic modeling is extremely important when you're doing this analysis. You cannot just rely on the direct energy, and the concentration can be pretty hard to predict because it's in a dynamic. So I'm not sure whether this makes more sense.
Well, I 100% agree with you that the kinetics are what drive the overall process, especially for these branching networks. You showed, I mean, all of us show these networks where A goes to 100 different products and through many steps. Not all of them will be equally populated out at the edges of the path. And it's kinetics that drive those things. Catalysts will have a huge impact as well. the mineral catalyst, for example, or the protonated forms pH catalysts. Anyway, very nice talk. And I think it's really fascinating to see how the different components that drive kinetics influence the outcomes. Thank you for
That's
that.
true. Yeah, thanks for the question.
Great. So I had a question for Bruno, and so my background is more in the geosciences, and I noticed that I think in your database you were saying that you might need some more geochemical or geological data inputs, and I was just kind of wondering what kind of data are you interested in, and do you have ideas on how you're going to kind of get the word out to different disciplines to kind of populate some of the different fields in your database?
Yeah, absolutely. The thing is, like, the way I see it in the end, well, we could just go narrowly to try to only put whatever that is purely prebiotic here. But if this really wants to be, if this really becomes like a useful resource for future origins of life researchers, I think it should also include like the environment. And in that sense, I think that both atmospheric chemistry and geochemistry are very important. So far, I'm not a geochemist. So right now, for instance, I'm being like just annotating what I read and what I understand, but I will be very good that like proper geochemists will also like make a little bit of effort. I'm not 100% sure like how, if geochemists already have like their own database. I know that there are like some programs that encode like a lot of reactions, and I've been looking at those like as a possibility to include them here. But yeah, so far in the end, like everybody that is interested on this discipline and has something to offer, it will be like welcome to include information, because this channel is actually if there's like a community behind putting information.
Great. Yeah, I think it sounds like it could become a really nice tool for everyone to kind of contribute and use.
The only thing I have to do is to make it fun, because otherwise it's going to be a little bit too much to ask. But anyway.
Yeah, I guess that's true. Like, people have to make the efforts to contribute. So if it's fun, then I think that would help a lot. So currently, I don't see any more questions in the chat or any raised hands. So maybe I'll just take a few moments to wait and see if anything else pops up. Or actually, something I think just popped in. So there was a question from Guillermo Bracamonte, and it was, how did you fix the initial conditions related with prebiotic chemistry? My question maybe is focused on the interval of time of the development of life where reactions were developed due to the interval could fix very different conditions. Yeah, so I'm not sure who it's directed to, but if either of you had any comments on that.
Well, from my perspective, I think that this question is not for me, but well, the only thing I can say is like one of the reasons why I think that we need a specialized database is because in the end, in some other fields, like for instance, organic chemistry, is very important to know when you have to add that reagent, when you have to condensate, when you have to do this and that. And in this case, it's all about conditions. So I'm super concerned about those. It's really like the thing that I want to catch with this database. But I'm pretty sure like this question is not for me. So sorry.
To fix the initial, I'm not pretty sure whether this is about the setting up the cytokinetic modeling or about the reaction exploration. But to provide a brief answer, like to the microkinetic modeling, we just provide the initial concentration and give it temperature, constant temperature and pressure during the simulation to see the evolution of those different species. And for the reaction network exploration, it's not related with the initial condition, which is starting from water and hydrogen cyanide, and each step with a new intermediate we continue add, considering reaction of unimolecular transformation and also bimolecular reaction with water and hydrogen cyanide. That's how we built up this network.
Okay, great, thanks. So I guess if we don't see any more questions or raised hands, maybe we can all go ahead and thank both speakers again. So thank you, Qiyuan and Bruno, for the great talks. And also thank you to everyone who tuned in to participate. And yeah, we'll be back in 3 weeks for another seminar. So we hope to see you again. And we look forward to also the life seminar series. So I guess, thanks, everybody.
Thank you. Thank you for the invitation. Thank you for listening.
Thanks you all.